We need a new phone. He's like, we're gonna protect this one because we've already broken like 6. So on this stage.
Luke, can you hear me all right?
Yeah,
How you feeling?
I feel all right. I feel like it's actually getting worse.
Okay. Okay. Okay, we're going to start here pretty soon, so.
The central nervous system? Me too.
Okay, now we're ready to start. All right, so let's have a word of prayer, then we'll begin. Dear Lord, it is your will that through our good lives, we would silence the ignorant talk of foolish people. We pray that we would live as free people, but not use our freedom as a cover-up for evil, but live as slaves of you. Lord, help us to show proper respect to others, to show love for the family of believers, to fear God, and to honor those in charge. Help us now honor the subject of human physiology. In Jesus' name, amen. Okay, we saw last time that the uptake of materials can sometimes be done without the expenditure of energy. But the universe of molecules that can enter the cell in that manner is pretty small. So it is your very, very small polar molecules like water. It's also small nonpolar molecules, usually flat aromatic molecules that can get through membranes. And then there are some molecules that can be taken up by facilitated fusion. But in human cells, number of molecules that can be taken up by in that fashion, you could probably list them on a 3x5 card you have in your hot little hand. It's not very many. So most of the things that you want to take up will require the expenditure of energy, and that process is called active transport. Now, diffusion, facilitated diffusion, passive diffusion, those processes are concentration dependent. Active transport is not concentration dependent. Because you're using energy, you can pump things into the cell or kick things out of the cell against a concentration gradient. That's one of the beauties of active transport. Active transport is essential for the life of the cell. The cell essentially donates a good proportion of its energy towards active transport. It has to do that in order to maintain its internal and external environment. So that's why active transport ends up being really quite important. There's two main types of active transport. They're so-called primary active transport. And in this case, you have a transporter. These transporters are invariably all embedded in the plasma membrane. The energy that they utilize comes from the direct hydrolysis of ATP. The direct hydrolysis of ATP. The direct hydrolysis of ATP will bring whatever you want to bring into the cell or kick it out of the cell against its concentration gradient. We'll look at some examples of primary active transporters. The one that's definitely the best studied is the sodium potassium ATPase. So this particular molecule has been extensively worked on. Jens Skouns, who was at the University of Oslo for many years, was a structural biochemist who did really, really nice mechanistic, physiological, and then structural work on this molecule. This is a molecule, a protein that's found throughout not just the animal kingdom, but also the plant kingdom as well. So this is a very, very, very widely distributed protein. This is an active transporter. It brings 3 sodium into the cell and exports 2 potassium from the cell. So I got that completely backwards, I'm sorry. It kicks 3 sodium out of the cell and brings 2 potassium into the cell. I caught myself, good thing I caught myself. So 3 out, 2 in, essentially. And because it's pumping against a concentration gradient, it will utilize energy to do that. And that energy involves direct ATP hydrolysis. Now think about this with me for a second. If you get, say, a blood test and your blood is dripped into the analyzer and all the components come up, what is the ion that is the most numerous in your blood plasma? Sodium. Sodium, because your cell is constantly exported from the cell into the plasma. What ion is found at very low concentrations inside plasma? Potassium, because most of your potassium is in your cells. Therefore, if you get a crushing injury to muscle, or bone for that matter too, damaged cells release lots of potassium. This release of potassium causes a condition called hyperkalemia. And that is one of the threats you have with a crushing injury. That's why with a crushing injury, you not only want to make sure that you don't have bits of marrow or something like that that have gotten into the circulation and can cause a heart attack or a stroke or something like that, but you want to also make sure that potassium levels essentially are brought down. You know what's one of the great ways to bring potassium down really quickly? Insulin. Insulin. Insulin injections bring glucose into cells, but they also bring a lot of potassium into cells too. And that's one of the treatments for hyperkalemia, insulin. And that will drop the potassium down too. In fact, your missus, who has to take insulin, one of the warnings on her insulin is probably hypokalemia. You take too much insulin because all your blood potassium goes into the cells, and now your blood levels of potassium become abnormally low. Someone had their hand up. Bracken, was it you?
It was me.
It was you,
My
yeah.
question was just gonna be that just means
Yeah,
high potassium.
right. Too much potassium, too little potassium. Now, unfortunately, this video doesn't work anymore because the new PowerPoint Word won't play it. So if you click this, it just goes here. So essentially, you bind to high affinity sodium sites. So I'll just use the picture here. And there it is. You bind to 3 high affinity sodium sites when the protein is in this conformation. When you bind to those, notice these are facing inside the cell. So the sodium has to come from inside the cell. You bind to these 3 sites and then the laser breaks. And when you do that, there is a aspartic acid residue here that now receives a phosphate from a bound ATP. That bound ATP is activated to phosphorylate the aspartate residue when these 3 sites are occupied. When the aspartic acid residue is phosphorylated, this protein now changes conformation. These sites will now release their sodium to the outside of the cell. And then these high affinity potassium sites now become available once this conformational change, these are now low affinity sodium sites. These will bind potassium from the outside of the cell. When they do that, the phosphate on the aspartic acid residue is hydrolyzed, now changes conformation back to the original conformation. This is now a low affinity potassium site. Potassium dissociates into the cell, and now you're ready to rock and roll all over again. High affinity potassium, sodium sites are remade. You bind sodium, the ATP binds, phosphorylates aspartic acid residue, changes conformation. Out you go, in you go. Hydrolyze the phosphate, Bob's your uncle, and then you repeat, rinse, wash, repeat, pass, go, collect $200, and there you are. This is an extremely active protein. 20 to 25% of the cell's ATP is dedicated to operating this thing. How many of you have heard of a rodent poison called rodent blocks? If you go to the church across the street, sometimes you'll see in the corners of some of the Sunday school rooms, you'll see these little wax cubes. Bethany, you've seen them? Those are called rodent blocks. They contain a drug called wabane that's embedded in wax. Rodents love wax. So they'll chew on the wax, but they'll also take up the wabane. Wabane poisons this. By poisoning this, it kills the little boogers and they go belly up. And then they're dead. And then you dispose of their disgusting bodies and you are out some rodents. So that's an example essentially of what happens when you poison, you inhibit this particular protein. That's how important it is to the life of the cell. No, just that one called wabane. There are other forms you could buy wabane in for around the house. It's those Orkin, when they come, they put those in your vents. They'll put these, they're not cylinders, they're not cubical, they're more rectangular. Yeah, they'll dump those into the vents because the animals will stay in the vents. In fact, you can hear them sometimes in the vents. Those essentially are wax containers that contain wabane. And they will chew on them, you know, bon appetit and buenas noches, raton. So,
Then do they crawl into the fence to get the animals out, or do they?
Yeah, doesn't it like
Yeah, they
just
just
like
sits
die
in the
there and
vent.
then it's like
yeah, and then you call Orkin and they come fish them out. And okay, some of you look at me as if to say, poor rodents. Oh, I'm so sorry for them. They're rodents. Get over it. If they're in my house, guess what? It's me or them. Now, the other option is you can get a cat. But getting my cat to go down the vent, that's a pretty tall order. So, come on, Oscar. Oscar, down you go. Shut up. Do your job. Yeah. Not going to happen. Okay. Those are primary active transporters. Direct hydrolysis of ATP drives transport. Secondary or coupled active transports use the energy found in concentration gradients. So when you have a concentration disparity on one side of the membrane as opposed to the other, that is an opportunity to do work. And you can use that to bring molecules into the cell, piggybacking them on molecules that will come in down their concentration gradient.
So is it still working against the concentration?
For one molecule, but not for the other. So one of the best studied of these, there are several of these that are well studied, but one that's very well studied is the sodium glucose co-transporter. So remember, because of the sodium potassium ATPase, sodium levels inside the cell are low. Sodium levels outside the cell are high. If you make it pour, sodium will come into the cell. Well, why not use the work that will generate to bring something in on the back of it? So the sodium glucose transporter finds a sodium and a glucose. And it brings in a sodium and a glucose. And then the sodium is just moved out by the sodium potassium ATPase. So you get a glucose using the energy stored in this concentration gradient. That's how secondary active transporters work.
So part of the reason why we put so much salt in the green glucose and stuff.
Someone salt in what?
Sports drinks. Like Gatorade.
Oh, sports drinks. That's what it said, Ford stream. Sports drinks, right. Yes, because you lose so much of it, and then it will help you take it up as well and get it into your body, so you can now do a lot more sex. Really wreck yourself. Yeah.
So in this example, the sodium is moving from high concentration to low concentration just by diffusion, and the glucose is moving against the concentration. Low concentration just by piggyback.
Exactly.
Does that require any not ATP, but any other sort of?
Not directly. There's no direct use of ATP. Primary active transporters use the ATP directly. Can't take you anywhere, can they? Do you guys see the Facebook post of softball players who are flexing?
Yeah, Kelsey.
Yeah, Kelsey in the back showing off them guns. Yeah, Kels. Yeah. Beast. I thought it was pretty cool. I said to myself, I don't know how much fun they had taking that picture. It was probably massive. And if that's the one they used, the others were probably pretty funny too. So there is no ATP used directly. It's just all concentration gradients. That's why it's secondary active transport. Now, you can have molecules that come in together, or you can have molecules that go opposite directions. You can also have other situations as well, but this type of situation here, where they come in together, that's called a symporter. And if you have molecules that go opposite direction, like for example, the chloride bicarbonate transporter, that will see, that's called an antiporter. These are both secondary active transporters. Just the directions the molecules move relative to each other are different.
The molecules are being transported, are they binding together, or are they just?
They bind to sites on the protein, the transport protein. The transport protein changes conformation and releases it into the cell. The transporter toggles between two different conformations. And that's actually a pretty interesting story in and of itself, but beyond the scope of this course, I'm afraid. Okay, this one worked earlier. Does it work now? And it's thinking, it's really thinking. I don't think it's going to work. I don't think it's going to work. So, okay, we'll just go on. All right, so the coupled transporters, they're symporters, they're antiporters. They're also known sometimes as collectively as co-transporters, but the mechanism they use is secondary active transport. No direct hydrolysis of ATP, they use the energy stored in concentration gradients. Now, cells can combine these in kind of a 1-2 punch sometimes. This is a pretty common strategy. You see this in the digestive system, you see this in the kidney, you see this in several different tissues, where polarized epithelial cells will have a co-transporter at the apical surface. They will bring in sodium and glucose together using a sodium glucose symporter. Then the sodium is removed into the bloodstream using a sodium potassium ATPase, which is a primary active transporter, but this is restricted to the basal membrane, whereas the secondary active transporter is restricted to the apical membrane. And then the glucose is removed via facilitated diffusion. Because glucose concentrations in the cell are high, because you bring it in, it can be kicked into the bloodstream rather easily, because facilitated diffusion works via concentration. So both of these molecules essentially are moved into the bloodstream. One, the sodium, by primary active transport, the glucose by facilitated diffusion. The facilitated diffusion transporter is restricted to the basolateral membrane, just like the sodium potassium pH. So by separating these in a polarized epithelial cell, you can use them together to get molecules from either the digestive system or the filtrate of the kidney or something like that to the bloodstream. Channels are pores in the membrane. They're not indiscriminate pores. They're pores that fit a particular ion, usually ion or molecule, very precisely. So precisely that only that particular ion can pass through that pore. So the channel is either open or shut because of a certain regulatory mechanism. But once you open it, the ion or the molecule or the whatever can pass through this according to the concentration gradient. So if we think essentially of this door as a channel, this doorway essentially will allow passage into the room. When it's too crowded in here, then people will start pressure. But we can regulate the flow by shutting the door. Shut the door, now you can't move things into the product. And that's essentially the way channels work. So channels, they're just openings in the membrane. The pores are protein-based pores in the membrane, but whether they're open or shut is a matter of regulation. We're going to see there are two main ways to regulate channels. One is by ligands, that is the binding of small molecules. The other is by voltage. The voltage drop across the membrane. When that changes, that will determine whether the pore will open or shut. And in fact, because you are pumping ions into the cell, from the cell, so on and so forth, you have a disparity of charges inside the cell relative to outside the cell. Typically, inside the cell will be more negatively charged than the outside of the cell. You're removing 3 sodiums, you're only replacing them with 2 potassiums. You're also pumping out calcium. Calcium concentrations inside the cell are very low. There's quite a bit of phosphate inside the cell. There's also protein. Protein on the average tends to be negatively charged. All that added together means the inside of the cell is more negatively charged than the outside of the cell. That creates an unequal distribution of charge across the membrane. Therefore, you have what's called a voltage drop across the membrane. This voltage drop can be measured. You can actually stick an electrode into cells and you can measure that charge difference across the membrane. If this sounds like it's hard to do, that's because it is. You need to make these electrodes. You need to make them essentially from pipettes. You need to pull the pipette very, 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 very thin. Then you need to break it. Then you need to polish it. Then you need to get a very fine needle in it. And then get the right solution in it. And then you need to take a cell and immobilize it. And then using a manipulating device, you need to get it so that you get it just inside the cell without exploding the cell. And it takes an extremely skilled pair of hands to do this and a whole lot of practice. So Nancy Diane O'Dowd was our patch clamper. She was very good at this. She could do this in her sleep. And I did rotation through her lab and I spent, 4 days. I think I spent 12 hours each day. And I think I got one of them patch clamps the whole time. And Diane kept, you're getting better at this. I said, oh, really? Could have fooled me. Anyway, there are some people who are very good at this. If you do this, you get a consistent measurement. That measurement is usually in the vicinity of about 70 millivolts for most cells. Now, some cells will vary. Cardiac cells, for example, cardiac muscle cells are about -90, but the rough average is about -70. And again, this disparity comes from the fact that you have such a wild difference in concentration between sodium inside the cell and outside the cell, potassium inside the cell and outside the cell, chloride both inside the cell and outside the cell, and calcium inside the cell and outside. Look at the calcium concentration inside the cell. Fleetingly small. That's due to the fact that you have a primary active transporter called the calcium pump that just spits out calcium all day. The other thing you have inside the cell that you don't have a lot outside the cell is phosphate. Phosphate's negatively charged, and then of course protein as well. So this is the case with the cell. Not only is this the case for humans, if you look at squid axons, boy, you see a trend that looks pretty similar. Lots of calcium inside the cell as opposed to outside the cell. Not much sodium inside the cell as opposed to outside the cell. Not much chloride inside the cell as opposed to outside the cell, and very little calcium inside the cell as opposed to outside the cell. So while the actual numbers differ, because these puppies live in seawater, no surprise there, the trends are the same. This, in fact, is actually kind of universal for animal cells and also to some degree plant cells. And the cell, again, expends a lot of energy to keep it this way. Now, some cells can vary the concentration of ions relative to each other and change that voltage drop across the cell, across the membrane. We call such cells excitable cells. Excitable cells. And excitable cells are found throughout the nervous system. We call them neurons. That's what we're going to talk about next. So if you take your average run-of-the-mill neuron, whatever that means, and you stick one of those electrodes into the membrane, you get a range for the resting potential of anywhere from -65 to -80 millivolts. Now, there are some neurons that will have resting potentials that are more than that. There are some that will be -85, but that's pretty much a rough average. In fact, if you look at just about any cell in your body, that is about what your resting potential will be, between -65 to -80 millivolts, with a rough average being about -70 or so millivolts. And it's due to the fact that you have lots of cations that have been pumped out of the cell. So certain cells in the body can change. There's concentration disparity across the membrane. That changes the membrane potential across the cell. These types of cells are excitable cells, also known in the nervous system as neurons. So neurons are going to vary their membrane potential. They do this by varying their permeability of their membranes to various ions, sodium first and potassium. So they create a small patch of membrane locally, whose membrane potential is different from the rest of the cell, but that small little patch of membrane that has a change in its membrane potential, that small little patch of membrane potential can spread to the rest of the cell, so that the whole cell undergoes this change in membrane potential. This wave of change in membrane potential, we call that an action potential. An action potential. So that sounds pretty cool, pretty neato. How do cells do this? They use something called the voltage-gated channel. Voltage-gated channel. So these voltage-gated channels are closed when a small patch of the membrane where that channel is located becomes more positive. They open. When they open, ions now will move into the cell and will exit from the cell. That will change the amplitude of the membrane potential, and that change in membrane potential will spread. So you have two main voltage-gated channels in neurons: voltage-gated sodium channels, voltage-gated potassium channels. Voltage-gated sodium channels, voltage-gated potassium channels. So here is a voltage-gated sodium channel, and here it is at rest. So at rest, you can see it is closed. As this little patch of membrane around it becomes more positive, it opens. Now, which way is sodium going to move relative to the cell based on what you know about sodium concentration? Is it going to leave the cell, or is it going to run into the cell? It's going to move into the cell. There's very little sodium inside the cell. Most of the sodium is outside the cell because of the activity of the sodium potassium ATPase. Sodium runs into the cell because you have these positive sodium ions running into the cell. That will make the membrane potential more positive. It will get to a certain point where an inactivation gate is going to essentially gag the channel and prevent more sodium from coming into the cell. And at this point, then the sodium channel will close and the inactivation gate will be removed like that. So that's this cycle. So this channel kind of opens, then, if you will, works itself out of a job and then cycles back to the closed version. All right, here's how an action potential works. We'll go over this, and I'll use a pictorial way of showing this to show you how it happens. And this is how it goes. So the cell is at rest. At rest, you have a membrane potential of somewhere around minus 70 millivolts. The cell receives some sort of stimulus. What that stimulus is will vary from cell to cell. We'll talk about what those stimuli are, but you're going to get a patch of membrane that will be depolarized up to about minus 55 millivolts. This value is called the threshold. At the threshold, the voltage-gated sodium channels are going to open. They'll open and sodium will diffuse into the cell. That will cause more of these channels to depolarize because it'll spread throughout the membrane. That causes more sodium to go into the cell. You have a positive feedback sort of situation. Eventually, you will depolarize the cell to somewhere around plus 10, plus 20 millivolts. At that point, the voltage-gated sodium channels are going to close and the voltage-gated potassium channels are going to open. Which way is potassium going to move? Will it move from the cell or will it move into the cell? It will move from the cell. Because there's lots of potassium inside the cell. Remember, damage the cell, you get hyperkalemia. So potassium moves out of the cell. You move from the cell, now the membrane potential is going to become more negative. It will then go back to resting, and the voltage-gated sodium channel, the voltage-gated potassium channels will now start to close. It actually overshoots it just a bit. And now the sodium potassium ATPase will return it to resting. And this essentially constitutes your action potential. So this first part, where the voltage-gated sodium channels are open and the cell now is becoming more, the membrane potential is becoming more positive. That is called depolarization. Depolarization. This is the work of the voltage-gated sodium channel. When it reaches its zenith, around about 25 to 30 millivolts, plus 35 millivolts, the voltage-gated sodium channels will start closing around here. When they're largely closed, and the voltage-gated potassium channels will start opening around here. They're now open, potassium now is going to leave the cell. Now you go back to normal, and this part of it is called repolarization. Repolarization. The neuron will usually overshoot just a bit, but that's okay because the voltage-gated potassium channels at this point are all shut. It works back. This portion of it is called hyperpolarization. And then you're back to resting potential. So let's use this smart art to show you how this works. At resting, everybody's closed. We have in the pink, the voltage-gated sodium channels, and in purple, the voltage-gated potassium channels. Watch them as they work. When you get to threshold, they're going to start to open. Sodium will now move into the cell. The membrane now becomes more positively charged. This is depolarization. The voltage-gated potassium channels open much more slowly. So it shows this one just starting to open. That's as it now starts to depolarize. It gets more positive. Eventually, the inactivation gate will shut, and these will now be open. So sodium is moving into the cell much faster than potassium is leaving. Eventually, repolarization, you can see that the inactivation gate is going to shut. Your voltage-gated potassium channels are now open. You're now going to repolarize. You're going to become more negative when it comes to your membrane potential. Potassium is leaving the cell. Eventually, they will hyperpolarize and they will start to shut. And then you'll reestablish the resting potential after hyperpolarization. And now you're back at resting. That doesn't work either. All right? Any questions about the action potential? So depolarization is associated with the activity of the voltage-gated sodium channel. Voltage-gated sodium channel. Repolarization is associated with the activity of the voltage-gated potassium channel. During depolarization, sodium enters the cell. During repolarization, potassium leaves the cell. And then at resting, everybody's closed and it's all IV. Okay, Evan?
Is there a reason during depolarization that so on that picture there was two no, just going back. Yeah, that one. So the one gate is open and the one gate is closed. Is that just the body knowing that it needs to bring in more sodium?
The potassium channels start to open during depolarization, but they open much more slowly than the potassium
So the one on
channels.
the right is not starting yet, correct?
Right.
Okay.
So your potassium channels will start, they usually open somewhere between -5 and +10. So that by the time you get to the zenith of your depolarization, the sodium channels are largely inactivated and then the voltage-gated potassium channels are open. Now, let's do a little bit of something that might interest you. How many of you have heard of a drug called lidocaine? Ever been sunburned? Ever put solar cane on your sunburn? you put lidocaine on your body then, because there's lidocaine and solaricaine. That's what kills the pain. That numbingness you have, that's lidocaine. Lidocaine is a local anesthetic. Ever been to the dentist? Had the injection? Now you know. My Libby's in my levy, so on and so forth. Ibuprofen, so on and so forth. You watch no comedy, and that's glorious. Lidocaine locally inactivates sodium channels. That's why you go completely numb. Because the sensory neurons now cannot form action potentials to convey sensory information. And so that's why it's used locally. You don't want that to go to your heart, though, right? That's why it's given locally and not intravenously. Because then you would die, yeah.
And so if they give you too much, that would spread.
It would spread, and that's why you never get it intravenously. Epidurals, that's with a drug called bupivacaine. Bupivacaine will very slowly diffuse from the cerebrospinal fluid, and as it diffuses, it gets broken down very gradually. If you give that one intravenously, it's very cardiotoxic.
So is a spinal block different than an epidural?
That's why I do an epidural is.
Yeah, they call them different things.
So Greg will get very savvy about giving those because dentists use a lot. In fact, another topical anesthetic the dentists use is topical cocaine. It's actually marketed as a mucosal anesthetic. It's a very good anesthetic. But of course, you just use it topically on the mucosa. And dentists actually use it. They keep it under lock and key. I know some dentists won't use it, but for numbing things that you need to do on the gum, it's extremely effective. Extremely effective. And it works in very small concentrations too. All right, that's a tidbit of information there for you. So the action potential begins with the depolarization of a little bit of the membrane, but that spreads. And it spreads throughout the whole neuronal membrane. It spreads down the neuron and all the way to the end of the neuron. And recall this action potential that propagates throughout the whole neuron. We call that a nerve impulse. A nerve impulse. And so the action potential essentially is going to start locally, but this local depolarization is going to spread. Now, it's only going to spread in one direction because once you depolarize, this part is just coming off its depolarization. So it's repolarizing, and it therefore is going to be resistant to depolarizing. So this will spread, and it's only going to spread in one direction. And this will spread essentially down the membrane like that. So if we go back, we can see it spreading down the membrane just like that. And that's essentially how you get this propagated. That doesn't work either. Right down the membrane like that. So neurons, as we said, they're not the most numerous cell in the central nervous system. Glial cells are, but they are the stars of the nervous system show. They vary pretty wildly in their size and shape, but structurally they have some similarities. That is, they have a cell body. The cell body has many extensions to it called dendrites. Dendrites will take nerve impulses that they receive from other neurons, and they will move those impulses toward the cell body, toward the axon. And then they have one axon. That axon may be branched, and sometimes can be very highly branched, but they only have one. And then this action potential will be propagated down the axon, be propagated away from the cell body towards the axon terminus, like that. And these branches of the axon are called collateral branches of the axon. This particular neuron here is called a multipolar neuron. A multipolar neuron. So it has a very recognizable cell body and an axon that you can definitely distinguish from the shorter and more numerous dendrites. And there are several important neurons that have this multipolar structure. This particular structure here, this is called a unipolar. A unipolar neuron because the axon and the dendrites look about the same. And distinguishing one from the other is really hard to do. There's a way to do it. You can actually load a dye in here called dye I, and the dye will be carried in the direction of the axon. So dye I, you can just you micro-inject it. Yes, that's hard to do. You micro-inject it into here, and so you inject it here, it'll go this way. You inject it here, it goes this way. And that's how you can tell which end is which. But structurally, these can be pretty hard. The axon termini are usually inflated. And sometimes these inflations are really subtle and hard to tell. So this makes it look really easy to tell. Yeah, it's not always so easy to tell. Now, this particular neuron, multipolar neuron, has an axon that is insulated with what's called a myelin sheath. A myelin sheath. And when you get an action potential that goes down a myelinated axon, the action potential actually is able to jump from here to here. This area where you don't have any myelin sheath, that's called a node. It's called a node of Ranvier, after the great French anatomist. And the node of Ranvier, essentially, you get your action potential going from here to here to here to here to here. That's called saltatory conduction. Saltatory conduction. And you might recognize the Latin root for jump, to jump. If you don't, you Spanish speakers might recognize the Spanish word for to jump in there. Saltar, to jump. Salta, salta, señor. So this is salta, 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 all the way to the end. So transmission through myelinated axons is much faster than it is through unmyelinated axons. Also, the thicker the myelination, the faster the transmission. All right, so here are your multipolar, bipolar, and then pseudo-unipolar. So notice with bipolar and the pseudo-unipolar, it's hard to tell which is which. Now with the pseudo-unipolar, by the way, your sensory neurons are pseudo-unipolar. And when you find them essentially in the dorsal root ganglia, that's what they look like. So these essentially have this, they have this neck essentially that feeds into the cell body, whereas the bipolar ones, you just get this glop into the, yeah, you don't have a neck, it just goes straight in there. So this has a neck, this is kind of one of those neckless people that looks like good dirt or something like that. And that's bipolar. Dendrites, again, they extend from the cell body. They're small, they're numerous, they bring impulses toward the cell body. The axon, there's one of them, but it can be highly branched, and it will transmit impulses away from the cell body. And the termination of the axon usually consists of a small inflation, albeit it can be very hard to see. So there are two types of channels. We already talked about voltage-gated channels, but there are also ligand-gated channels. Ligand-gated channels. Ligand-gated channels are small molecules that bind to a receptor site on the surface of the channel. When it binds, the channel changes conformation and it either opens or closes. So ligand-gated channels open in response to being bound by a small molecule. Nicotinic acetylcholine receptors found in muscle Those bind acetylcholine and they open. Those are ligand-gated channels. Voltage-gated sodium channel, voltage-gated potassium channel, so on and so forth. Those are voltage-gated channels. They open or shut according to the voltage drop, the amplitude of the voltage drop across the membrane. Excuse me. That went down the pipe. Now, your dendrites and your cell body of the neuron, they contain mainly ligand-gated channels. The axon, however, contains mainly voltage-gated channels. This is essential for the way the neuron is going to work. So the dendrites, the cell body, mainly ligand-gated channels. The axon, almost exclusively voltage-gated channels. We'll see why in just a bit. You're stealing my thunder. Now, what is the stimulus that the neuron will receive? Typically, the neuron is in some sort of structural communication between itself and another neuron. And another neuron will dump neurotransmitters that are going to flood the dendrites and cell body of that other neuron. So your stimulus is usually in the form of a neurotransmitter. Now, some neurotransmitters are stimulatory. They will stimulate this neuron. There are, however, some neurotransmitters that are inhibitory. They will tend to shut down the other neuron. So the stimulatory neurotransmitters will cause the opening of a ligand-gated channel. And this will create a local depolarization around the area that is receiving the neurotransmitter. What is the amplitude of that depolarization? Depends on how much ligand the dendrite and the cell body are exposed to. If they're exposed to a lot of it, then that localized depolarization can sum up to a proper threshold. That will cause the propagation of a action potential down the axon. So the type of depolarization that you get in the dendrite and cell body is of a different quality than what you see in the axon. What you get in the cell body and in the dendrite is called a graded potential. A graded potential. These potentials are local, but they're additive. They're additive. Will they add up to threshold? Depends how robust a response you get. And that response depends on how many of these ligand-gated channels get opened and how long they stay open. And that's dependent on how much ligand you have binding to them. They, in fact, do. That's how the axon knows whether or not to fire. All right, so graded potentials are local, but they're additive. And the summation of these can eventually get the cell to potential. So the place in the neuron that you need to get to this threshold of minus 55 millivolts is called the axon hillock. And the axon hillock is that area between the cell body and the axon itself. If you get the axon hillock to a threshold, the axon will undergo a proper depolarization. So here is a multipolar neuron. Here's the axon, here's the cell body. That's the axon hillock right there. That's the part you need to get to threshold, minus 55 millivolts. The input to the dendrites and cell body will be local, but it can spread and add all across this cell to eventually become minus 55 at the axon hillock. And if it does, then you initiate an action potential at the axon hillock. But my friends, the action potential in the axon is due to voltage gated channels. So that is not a graded potential in the axon. The depolarization that you get in the axon is what is known as an all or nothing. An all or nothing potential. You either get the axon hillock to threshold and you get an action potential, and the axon you don't, and you don't get an action potential going down the axon. That's why it's an all or nothing event, because it's a result of these voltage gated channels, lots of them. And once that gets depolarized, it propagates and you can't stop it. What happens when the action potential gets to the axon terminus? The axon terminus is slightly inflated, and it is known in the anatomical literature as the terminal bouton. Do I have any French speakers here? Bouton, does that mean anything here? I can always count on a Canadian, because Canadians have to study French. I don't have any Canadians, do I? Softball always has usually one or two Canadians. No Canadians. Anyone take French in high school? No? Or you forgot it all? Bouton is French for button. Button. Terminal button. So it's inflated, it's like a little button. If you call it the axon terminus, I will not get mad in the least. If you call it a terminal bouton, I just might buy you a coffee at Sacred Grounds, because that means you listen. Now, if you take a high resolution transmission electron microscope picture through a terminal bouton, here is the dendrite of the postsynaptic neuron. Here's the axon terminus of the presynaptic cell. Tell me what you see inside this axon terminus. Vesicles. Tons and tons and tons and tons of vesicles. Those vesicles are laden with neurotransmitter. So the action potential gets to the terminal bouton, and in the terminal bouton, you have voltage-gated calcium channels. Voltage-gated calcium channels. No. Calcitonin is a hormone that controls calcium levels inside the body. All of this debate about how much work calcitonin does, because mice that don't have calcitonin seem to be completely normal, based on the laboratory. I'll bet you if you release them into the wild, they probably die of thousand deaths. But that's another story. Yeah. Just because a white mouse is healthy in the laboratory may not mean squat, but who knows? Yeah, here, kitty, kitty. Yeah, no. Okay, well, story. We had to raise rodents because we had to inject them. We had to inject rats to get antibodies. So we use rats. They're cheaper than rabbits. And their antibodies are actually pretty good sometimes. So if we had a rat that was old that wasn't doing very well, we took it to the snake lab downstairs. And we said, I'm gonna feed you snake. And so we gave them the rats. And that way we never had to fill out an animal note as to what we did with the animal carcass, because it went in the belly of that python. And that python loved rats. Every time we got to the python got very happy. Okay, so calcium is going to cause an exocytosis event. You'll dump neurotransmitter into the space between these two neurons. The space is called the synaptic cleft. The synaptic cleft. And now those neurotransmitters will bind to their target ligand-gated receptors and causative potential in this photosynthetic cell. That's where we'll pick it up on Friday. Don't forget, we have a quiz on Friday. The quiz will be over transport, the transport chapter, which is chapter, wait, I've got my book right here. My book right here. Which is chapter 6. Chapter 6, okay? I'll give you a reprieve for chapter 5. And Luke, you can take, I can email you the test and you can take it and email it back to me. All right?
Okay.
Okay.
Yeah, that works.
All right. I will see you on the 14th, I guess.
Yes.
Stay well.
I tried to say it earlier, it's like